Welcome to the protein synthesis slideshow. This is absolutely my favorite thing to teach students, so I hope you guys love this concept as much as I do. During the protein synthesis unit, we're going to talk about what DNA does. We're going to talk about why DNA and RNA are both important. We'll talk about the differences between DNA and RNA, the different types of RNA. We'll talk about how proteins are made, and transcription and translation. And the big thing we're going to talk about is how to use a codon chart. So we've been talking about DNA this past week in class. DNA has a huge job in a cell. The DNA molecule is going to contain all of the hereditary information within a cell, and as a result, within an organism, and it's going to hold that information in the form of genes. If you take a look at the picture on the right, you will see bands, those darkened areas on the chromosome. Those darkened areas are pieces of DNA, which are called genes. Again, a gene is a section of DNA that is coded, and that means that it tells our cells how to build specific proteins that the organism needs. Genes are going to code for everything that the body needs or that the body does. So things like your saliva, your bones, your eye color, your eye shape, your hair texture, your skin color, all of those things are coded for by DNA, and they tell the organism to make a specific protein. So they tell your body to make a certain protein that controls those characteristics. Because DNA is huge, it is stuck inside the nucleus. When we talked about the nucleus, we talked about the fact that the nucleus is in the center of the cell, and it has small holes in it called nuclear pores. Make sure you write that down on your notes, because that's kind of important. Okay, so the nuclear pores are small, and DNA is big. DNA is actually stuck in the nucleus. It can't get through the pores. So we solved that issue by having a messenger that's able to go through the nuclear pore and carry the DNA's information out into the cytoplasm to the ribosomes. Remember that the ribosomes are the protein factories of the cell. So again, this messenger is going to take the message from the nucleus out to the ribosomes, where the proteins can be made. Earlier in the year, we talked about different types of nucleic acids. And you know already that DNA is a nucleic acid. The other type of nucleic acid is RNA. And we actually talked about another, but we're talking about RNA specifically in this unit. So RNA is a nucleic acid that is a messenger between DNA and the ribosomes. There are three key differences between DNA and RNA. RNA has a ribose sugar, whereas DNA has deoxyribose. RNA is single-stranded, whereas DNA is double-stranded. And here's something that's really important. Put a star on this next to your on your paper. Put a star next to this on your paper, sorry. RNA contains a nitrogen base called uracil instead of thymine. So in RNA, the base A pairs with U instead of T. That is a key thing to understand. Before we go on to this next piece, I want you to take a look at your note outline. You will see that there is a Venn diagram that asks you to compare RNA and DNA. So on the left side of the Venn diagram, you are telling about DNA. On the right side of the Venn diagram, you're telling about RNA. And in the middle, you're going to talk about things that both DNA and RNA have. So pause this video, fill out your Venn diagram, and we'll go over it in class. All right, so you filled out your Venn diagram. Let's go on to the 3 types of RNA. The first type of RNA is called messenger RNA. This is literally the message that the DNA is going to send out into the cytoplasm to the ribosome. Messenger RNA's job is to copy the DNA message in the nucleus and take that information out into the cytoplasm to the ribosome. Then we have ribosomal RNA, or rRNA. Ribosomal RNA makes up the ribosome. And the ribosome's job, and so therefore the ribosomal RNA's job, is to read and decode the messenger RNA, or the mRNA. And finally, we've got transfer RNA, or tRNA. Well, you know what the word transfer means, to move things from one place to another. And that's exactly what transfer RNA does. Transfer RNA is going to carry amino acids to the ribosome, where they'll be joined together to form proteins. So transfer RNA is actually going to hold on to an amino acid on one end and some bases, some nitrogenous bases, on the other end, and it's going to bring that amino acid to the ribosome to help form that protein. More about that later. If you look at the bottom of the slide, you'll see a picture of messenger RNA, which is single-stranded and small. It is small enough to fit through that nuclear pore and take the DNA's message out into the cytoplasm. You have ribosomal RNA, which makes up the ribosome. And finally, you have transfer RNA. And notice, if you look at that picture, you'll see that at the bottom there's an anticodon, which is a set of bases. And at the top, there is that red piece is where the amino acid will attach. One of the things that I love to ask students on quizzes and tests are to describe the central dogma. And I think that's important because you guys will read that word, those words on tests, or hear that word, and it sounds complicated, but it's not. So I want you to really understand what the central dogma is. And you might want to jot this down on your notes. The central dogma simply says that we take the DNA's message, we transcribe it to RNA, and we translate it into protein. It's really simple. So we go from DNA to RNA to protein. Make sure you have that written down because I am going to ask you about it. Protein synthesis is actually the assembly of amino acids by RNA into proteins. And just like I said when I mentioned the central dogma, protein synthesis has 2 steps. The first step is transcription, which is copying the DNA code into messenger RNA. And then there's translation, and it's where you the messenger RNA's code is read, and then the transfer RNA helps to assemble that code into a polypeptide chain or a protein. Remember that the monomer of a protein is an amino acid, so that polypeptide or that protein is going to be made up of amino acids. So the first step of protein synthesis is called transcription. This is performed in the nucleus by messenger RNA. Remember that I said the DNA is too big to get out of the nucleus. The nucleus has holes in it called nuclear pores, but the DNA just can't fit through the hole. So this is where that DNA actually has its message turned in to messenger RNA. What happens is the messenger RNA is going to read a single DNA strand, and it's going to form the complementary copy. We're going to do some work with this in class, and remember that instead of having the letter T or thymine as a base, the messenger RNA will have U or uracil as a base. So when we read that DNA strand, every time we see an A, instead of pairing with a T on the RNA, that A will pair with a U. And we're going to do some practice with that again in class. So how exactly does transcription work? Well, first, the DNA strand is going to split apart, and it's going to expose the active strand, or the strand that we're going to form an mRNA complement for. Next, complementary messenger RNA nucleotides, including U or uracil, not T or thymine, will line up opposite that active strand, and they're going to form the messenger RNA. So if you look over at the picture on the right, you will see a pink messenger RNA strand being formed. Once that strand is formed, the messenger RNA is free to leave the nucleus through the nuclear pore. Next, we have translation, which occurs at the ribosome in the cytoplasm. If you remember back to cell structure, you know that we have ribosomes in two places in the cell. And make sure you write this down too on your notes. So the ribosomes that you have will either be found in the cytoplasm or bound to the rough endoplasmic reticulum, or the rough ER. Well, translation is going to happen typically at the ribosomes that are free ribosomes, or floating around in the cytoplasm. It is during this step that all three types of RNA will work together to produce proteins. And I usually ask that on a quiz. Another thing that I like to ask on quizzes is why all cells need ribosomes. And the answer is right here on this slide. All cells, even prokaryotic cells, need proteins. So in order for them to make proteins, they have to have ribosomes. So all cells, prokaryotic or eukaryotic, will have ribosomes, free ribosomes, floating in their cytoplasm. So this is how we take this messenger RNA and we make it into a protein. What happens is we have the sequence of bases on messenger RNA, which are going to serve as instructions for what order we want to join the amino acids together to make the protein or the polypeptide. So the mRNA will tell the cell that they need the amino acids in a certain order, and that's the order that they'll be made into a protein in. So ribosomes' jobs are to decode the instructions by using codons, which are a set of 3 bases on the messenger RNA that code for one amino acid. When we were doing our DNA, I got ahead of myself and I said that a codon could be found on the DNA. That's not quite true. Codons are actually found on the messenger RNA. I do like to give you DNA bases in sets of 3 to make getting your mRNA codons easier, but codons are actually on the messenger RNA. Each codon is going to be matched with an anticodon, and that anticodon is a complementary sequence that's going to be on the tRNA that will determine the order of amino acids. Okay, so that anticodon is on the tRNA. And the anticodon is going to be at the bottom of the tRNA. And at the top, there will be the amino acid. And I'll show you some more examples of this in class. Here's a picture that kind of shows the example. So if you look at the red line at the bottom of the picture, you will see that that's an mRNA molecule, and it has the codons, or set of 3 bases, on it. The tRNA looks like a letter T, and they're black. They're actually at the top. You'll see that at the bottom they have an anticodon, and notice that there are no thymine bases because this is RNA. So we have U, or uracil, instead of thymine. So the anticodon is at the bottom, it's in blue. And then at the very top of the tRNA, you see a red box, which is, for example, holding the amino acid serine. And then you see a green triangle that's holding the amino acid tyrosine. And we're going to talk about how we determine which amino acid should be on the transfer RNA. And that's something that I'll be showing you guys more of in class. It's hard to do over a video. Here's the information that I will give you about using a codon chart in this slideshow. A codon chart is used to determine the sequence of amino acids in the polypeptide. And we typically will use the one at the bottom that's like a box, although the circular one has been given before on the air test, so we'll learn how to use both, but I prefer that you use the one that's a box. So the codon chart uses the set of 3 RNA bases, or the codons, to find the corresponding amino acid that's going to be brought to the ribosome by the tRNA. More about this later in class. Just take note of the fact that one codon gives you 1 amino acid. So that's pretty much all I'm going to say about codons today.